Welcome everyone to our MStranslate interview today. It's our pleasure to be joined by Dr. Fred Sedel, who is the CEO of Medday Pharmaceuticals. And you may have heard of Medday Pharmaceuticals recently. They sort of shot to fame last year when there was quite a bit of media coverage of the high-dose biotin therapies that were gaining a lot of attention, and in particular their compound MD1003 that Dr. Sedel is going to be talking to us about today. So, Dr. Sedel, I wonder if you wouldn't mind starting just telling us a little bit about Medday, to begin with MD1003 and how that project came into existence.
Yes, so Medday is a company which develops drugs for neurological diseases, and these drugs have the specificity that they target the brain metabolism. So this approach comes from an academic project initially, in which I was involved, and which was to identify new metabolic diseases in adults with psychiatric or neurological disorders. So by using this approach, we realized that targeting brain metabolism could have very significant effects in some patients who have so-called degenerative disease. And this is what gave us the idea to create Medday. So the originality of this approach is that from inborn errors on rare disease, we are very much patient-focused in the sense that the drugs we develop has to demonstrate a very significant effect in small numbers of patients, which is the way we usually develop drugs when you have only few patients to treat the drug. And this is the approach that we try to apply for more frequent neurological diseases, including MS. And I would say that we were a little bit lucky in some aspects to find that this approach could work in progressive MS, but as we'll speak later, the data seem very convincing.
Well, often the best research does come about from luck, doesn't it? So, you know, it always seems to be the way. So talking about MS, there were two studies in particular that you reported on recently, the MS-SPI study and the MS-ON studies. Can you tell us a little bit more about those studies?
Yeah, so the MS-SPI study is a study which is really addressing the potential of this high-dose biotin to treat patients with progressive MS. And we have selected in this trial a very classical population of patients who had progressive walking disability during the past 2 years and who entered the trial to be treated with the drug. MS-ON is targeting a different population. We have in this trial both patients with what we call progressive MS, but also patients with relapsing remitting MS. And the idea behind the MS-ON trial was to see whether the drug would work only in patients with progressive MS, or whether the drug could also increase the recovery in patients who have non-progressive sequelae after an acute relapse, which was a different question. So the MS-SPI really addresses the progressive MS population, whereas the MS-ON study addresses both.
Okay.
That's the main between the two.
Fantastic. And so I know you reported a little bit on this at the recent American Academy of Neurology conference. Can you tell us a little bit about the key highlights of those studies, the findings that you've obtained?
Yeah, so we now get the full data of both studies, and I think that the data are quite clear. For the MS-SPI study, the first thing we see is that there is, as a mean, it seems that the progression of the disease has been very much delayed or stopped in the population who have received the drug, and this is maintained over 2 years. But on top of that, we see in a significant proportion of patients, in a subset of patients, but which is statistically significant, what we call a reversion of progression, meaning that the patients are better after 1 year of treatment, and this is confirmed after 2 years of treatment, than they were before starting the drug. Okay, so this is the improvement of this patient. So that's the results of the MS-SPI study, which clearly show a benefit in the progressive MS population. For the MStranslate study, I think that we have the same trend, which is observed only in the population of patients with progressive MS, which was included in the MStranslate trial. But on the other hand, we did not observe any effect in this second trial in the population who had non-progressive sequelae after an acute relapse, which answers in some way the question that I raised earlier, which was whether the drug would work in both cases. It seems that it works only in the progressive MS population, but not in the acceleration of recovery after a relapse.
Okay, and it's really interesting that you. I mean, it's really interesting to have those results for progressive patients in both studies and see that it was specific to them. And obviously extremely exciting considering we don't currently have any available medications for people that specifically work in progressive MS.
Yeah.
So I know originally you mentioned that you were looking at brain metabolism and treatments that could work on that. Do you have an idea as to how the concentrated biotin might be working to have these effects in people with progressive MS?
Yeah, so we start to collect data from the trials first, but also from animal studies which are ongoing or in vitro experiments. And if you want, in progressive MS, our view is that there are three different kind of mechanisms which are responsible for progressive MS. First, there might be some inflammation, which is still ongoing in progressive MS. Second, there is chronic demyelination, which is obviously very important. And third, there is some what we call axonal degeneration. Okay, so these are the three parameters which are responsible for progressive MS. So what we know from the studies and from the data that we have collected is first that we have no effect at all on inflammation. So that's not the cause of the effect. Second, obviously there is very clear evidence for some neuroprotective effect. So we think that the effect that we observe goes through axonal protection or protection against axonal degeneration, I would say. And then for the remyelination or for the chronic demyelination, we are still working on that. I would say that we do not have yet very firm proof that it works through remyelination, but the experiments are still ongoing. So this is what we have in hand for now.
Okay, that's really exciting. So I guess one of the key questions that anyone asks, and I guess people are really interested in when we're looking at new potential treatments and clinical trials, were there any adverse safety events, any safety concerns that people experienced during these trials?
Yes, there are. So as a drug development, there are always some safety concerns or some potential safety concerns. The first one is that we observe some teratogenicity, malformations, fetal malformations in some animal species. And this implies that patients, women, childbearing women or women who are at the age where they could be pregnant, have to use a powerful contraception to avoid pregnancy. So that's the first point. The second one is that we observe that this high dose of biotin could interfere with some biological tests which are using biotin as a reagent. And this may cause some diagnostic errors. For instance, in the trials, we observed few patients who had false hyperthyroidism. Okay, so biological signs of hyperthyroidism, which were not related to any clinical symptoms of hyperthyroidism. And we realized at some point that it was false results caused by the very high level of biotin in the blood. And other biological tests may be concerned by these interferences. So that's the second point. The third one is that biotin may interfere with glucose metabolism, and diabetic patients who are using insulin or hypoglycemic drugs may have some dysregulation of their glycemia with a hypoglycemic episode, which can be a concern. And the last point, which is not directly related to our trial, is the fact that we are using very high dose of biotin. And so there is some concern that non-pharmaceutical grade biotin, because we know that people are treating themselves with some different sources of biotin, may contain some impurities that may cause some safety issues or safety concern in the long run. So that's another thing that people have to look at in the long term.
Okay. All right. So, I mean, there's a few there, but they seem like now, having picked them up during these clinical trials and understanding them, that they could be relatively easily managed if this was to progress further as a treatment.
Sorry, can you?
So it seems as though even though there was a few safety concerns, but now understanding these concerns, they can all be relatively easily managed if this was to progress further.
Yeah, so far, you're right. But remember, keep in mind that the trials were relatively small compared to other big studies in MS. There were only 250 patients, if we include the two trials, who were treated for at least 1 year. So that's relatively small population to really address all the potential adverse events. So it means that this is still a drug under development. It's not the end of the story. We don't know exactly what will happen in the longer term, I mean, if we treat more patients. So that's why everything has to be very carefully monitored during clinical trials, using different tools like registries, etc., to record the adverse events. And it will be at the end the decision and the responsibility of the health authorities to authorize or not the use of this drug for a larger population based on the existing data, but also based on prospectively collected additional safety data. So for now, we cannot answer completely to this question. Obviously, it's not a physiological dose of biotin that we are using. It's 10,000 times the dose of a coenzyme, which is targeting very important metabolic pathways. So we don't know exactly what it will cause at the end. So far, so good, I would say, but we have to remain cautious about that.
Okay, fantastic. So we had a few questions sent in by members of our community. So I'll just ask you a couple of those now. So one of the questions was about the protocol that was used in the studies where the placebo group was switched on to the high-dose biotin after the first year of the study. So the question was basically, what was the rationale, or what was the reason for doing that? And do you think that impacted on the results at all?
Yeah, so the reason is that when you switch the placebo to the active, that's a way to assess the effect of the drug in the same population. That's the same patients who are first on placebo on the drug, and then on the drug, which is different than comparing two different populations, which was the case in the placebo control phase of the study. That's the first point. The second one is that you can compare patients who have received the drug for 12 months at the end of the extension phase, which are the placebo who have been switched for 12 months to the active drug, to patients who have received the drug for 2 years. And this is blindly because nobody knows in the study, even during the extension phase, whether they have received or not the drug during the first part of the study. So that's a way to answer the question whether delaying treatment onset may have an impact on disease progression or not. And I think we have quite clear data from the MS-5 trial, which shows that although the placebo still benefited from the drug during the second part, which is the confirmation of the efficacy of the drug, these patients remain at a higher level of disability, okay, which means that probably there has been a disease-modifying effect in the patients who have been treated since the beginning, because these ones remain at a lower level of disability at the end of the extension phase.
Mm-hmm. And correct me if I'm wrong with this, but within the first 12 months of that study, when you did have treatment versus placebo alone, you did see an effect in the treatment group, didn't you?
Yes, and the results were statistically significant. So the primary endpoint, which was the reversion of progression, was statistically key with what we call the p-value, which should be below 0.05, but which was here at 0.005, so 10 times less than the required threshold. But other secondary endpoints, which are more, here to tune the effect and to see the effect of the drug on other dimensions, were also statistically significant. So yes, the trial, the placebo-controlled phase, showed a clear benefit of the drug over placebo.
Okay, fantastic. So the next couple of questions are linked, and I think come based off some information that was released when these results were reported, which mentioned the potential mechanism as being involved in part of the Krebs cycle. And so the Krebs cycle is part of the process that leads to the production of energy. So one of the questions was whether or not, well, firstly, what form of biotin you'd used, what specific form in terms of different stereoisomers. Obviously, someone with a chemistry background asking that question. And whether or not you'd looked at combining biotin with any other compounds that were believed to assist in the Krebs cycle.
Yeah, so the biotin is the biotin, which is the common form, the common isomer. The difference here is that it's an extra pure pharmaceutical grade biotin. So there is no single impurity in this biotin. It's what we call GMP biotin, so using the good manufacturing practice. And this is not, as we know, the case with other sorts of biotin, which are not necessarily pharmaceutical grade. They are okay for food supplements, but they are perhaps not okay at this very high level. So I don't think that it impacts very much the efficacy, but it can impact the safety. So we know that we are using only biotin. There are not chemical intermediates in what we give to the patient. So that's the first point. The second one, I think it's a good idea to combine, and we are exploring that. But up to now, we only have tested biotin itself.
Okay. And the follow-up question to that was, is there any idea as to why that Krebs cycle might be impaired in people with MS?
Yeah, so what we know in MS is the fact that there is an impairment of some mitochondria. So there are bit more mitochondria in the cells, in the neurons of patients with MS, but with time, many of these mitochondria become impaired, and the Krebs cycle lie into the mitochondria. So in our view, it's still a working hypothesis, but what we think is that biotin may support the healthy mitochondria within a group of mitochondria. Some are not functional, others are. And probably we can boost the Krebs cycle in the mitochondria which are still functional. And there is still a possibility that biotin may help also to generate additional mitochondria. So we are working on that. We have obtained some preliminary data that suggests that it might be the case, but this has to be confirmed. So these are the main hypotheses.
All right, fantastic. So as the last question, I guess, for people with MS out there who are watching this video, should they be cautious about this research? Should they be excited? Should they be optimistic? I think you've already answered my second part of this question, which was that people shouldn't be rushing out to get themselves biotin supplements, that there could be safety issues associated with doing that. But about the research in general, excited?
Yeah, I think they can legitimately be excited. I sincerely think that it's a real hope for patients with progressive MS. But as you just say, in the same time, they have to remain very cautious, and I'm speaking also about the physicians, because it's not the end of the story. We have to collect more data. It's still a product under development. There will be another international trials. We have, I think, good data so far. We have good safety signals, but here again, it must be the decision of the health authorities to assess the benefit and risk ratio based on what we can generate, and also to regulate the use of this biotin, because it's no longer a food supplement. Because of the very high dose we give, it becomes a drug. So everybody has to remind that. I can understand that many patients have want to take it and are in a kind of emergency, but I think it would be important to wait a little bit more. I don't know how much, or perhaps 1 year, 2 years. We are doing our best to get the drug to the patient as fast as possible. Mm-hmm. But I think we have in principle to remain a little bit cautious about it.
Okay. And so just currently, are the larger studies underway? The next studies, are they underway? Are you currently recruiting for those studies?
Yeah, so this will be announced soon. We are preparing for the moment for this study, so it's not yet ongoing. The protocol is not totally finalized, but I can tell you that this is something we are looking for. I hope end of this year, so it should not be too long at the end to get the drug approved. This is what I hope sincerely.
Okay, well, it goes without saying that we are extremely appreciative of you giving up your time today to give this interview and to tell us more about this hugely exciting project. I know that not only us, but everyone watching this video and people with MS around the world are extremely thankful and extremely appreciative. Very excited about the research that you're doing, and we'll make sure that we keep them updated when those larger trials begin and if they can potentially play a part. But good luck with all of the future research. We are all very excited, and thank you again.
Thank you very much, Fred. It was a pleasure.
Thank you.
Bye, everybody. Bye. Thank you.